So thank you for the invitation, much appreciated. This is a slightly different audience for me, so I'll try and introduce the background to this. Some of you will be aware of the context, some of you may not be, so if this is all old news to you, then I apologise. But the context to this question of the extent to which the structures that we work within, and in particular the incentive structures that we work within, shape our behaviour as scientists, I think is a really topical one. And it's exemplified by these recent papers which describe what has become known as the reproducibility crisis. So this question of the extent to which research findings, in particular in the biomedical sciences, but perhaps not exclusively, may not be as robust and reproducible as we might hope. So we have converging evidence from multiple domains that there is something here that we may want to look at. We have evidence from the pharmaceutical industry, exemplified here by this paper on the bottom left, that when they take research that has been generated by academic institutions and try and replicate those findings in their drug discovery programs in order to identify targets to take forward into further research, they're not able to do so in the majority of cases, and something like 60% of the findings from the academic literature can't be reproduced by the pharmaceutical industry. And as a result of these growing concerns over the last maybe 10 years, there have been efforts to attempt to empirically estimate what proportion of the published literature is actually robust and reproducible. So we have, for example, the cancer biology reproducibility project at the top, and at the bottom right, the psychology reproducibility project, which published its results recently. There's no particular reason, I don't think, to believe that one field is any better or worse than the other necessarily, but I'll come back to what we can learn from different fields in a moment and how that has a bearing on the open science agenda. I think one of the reasons why psychologists in particular have become interested in this question is that if we're right that what is distorting the value of science is the extent to which the incentive structures that we work within shape our behaviour as scientists, then ultimately that's a question of human behaviour, and you would expect psychologists to be interested in that kind of thing. So what did the Reproducibility Project Psychology find? Here you have two figures. The one on the left shows the p-values from original studies and replication studies, and the one on the right shows the effect sizes from original studies and replication studies. And there are two headline messages here. The first is that nearly all, over 99% I think, of the original, about 99% of the original studies reported p-values less than 0.05, what is conventionally described as statistically significant. That's the filter that you have to pass in order to get something published in an academic journal for the most part. And the extent to which that's the case is patterned by different disciplines, and psychology is one of those disciplines where there really is a strong emphasis on novelty, discovery, finding something and publishing something with a p-value smaller than 0.05. But when we look at the replication studies, the p-values are much more evenly distributed, and what that means is that many of those original findings simply couldn't be reproduced, at least with the same strength of statistical evidence. When we look at the effect sizes on the right-hand side, you see a similar pattern, but here in the opposite direction. The original studies tended to suggest quite large effects, partly because they had to cross that filter in order to be published, and that tends to inflate effect size estimates. And when we look again in a replication study, those effect size estimates tend to be smaller. That doesn't mean that those effects aren't real, and we can't say that just because there's been a single failure to replicate that effect is non-existent, but it suggests that many of the studies that we see in the published literature may not be robust, and at the very least they're probably telling us that effects are larger than they actually are in nature. So what's the source of this problem? And is it in fact a problem? Because describing it as a crisis is perhaps too extreme, because this really is an opportunity for us to reflect on the way in which we do science, reflect on the tools that we use to do science, and consider whether or not we need to bring any of those processes up to date and reshape the incentive structures that we work within. And again, that's a point I'll come back to in the context of the open science agenda. So we all know what we should be doing. We should all try to be the platonic ideal of a scientist where we are completely impartial, not invested in the results of our own experiments. We simply observe nature and describe it as accurately as we can do, and design experiments to test hypotheses that arise out of those observations. But the reality may be slightly different, because we need to get grants in order to grow our research groups. We need to publish papers in order to get promoted or move on to more glamorous institutions. So the things that we're rewarded for are not necessarily the things that we should be rewarded for if we want to be objective, impartial scientists. In other words, we're very rarely, as scientists, rewarded for being right. We're rewarded for publishing, we're rewarded for getting grants, we're rewarded for getting into certain kinds of journals, but none of those actually align particularly well with our results being correct. So maybe the scientific method has become distorted by the incentive structures, the scientific ecosystem, if you like, that we work within. So the blog in Euroskeptic has described what this looks like, and it's essentially a continuum of behaviour. So this obviously borrows from Dante's Inferno, and you have these different levels of scientific behaviour which are, if you like, questionable. So it's not fraud. There are cases of fraud, but that's not really what we're talking about here. It's the extent to which you oversell your data, you enthusiastically interrogate your data to get a result which perhaps aligns with your preconceptions, the extent to which you are more likely to error check your data if your first pass analysis doesn't tell you what you were looking for, as opposed to if it does. So you have overselling, post hoc storytelling, p-value phishing, all the way down into fraudulent behaviour, falsification in the icy jaws of Satan in the depths of that pit. What does that look like in practice? Well, there was a nice paper by Simmons and colleagues a couple of years ago, which collected real data and showed that if you were randomised to listen to a certain kind of music, you became younger. Not you felt younger, you became younger. And this was an experimental study, so it was causal. It meant that listening to that kind of music turned back the arrow of time. How could they arrive at such a clearly false conclusion? I assume it's false. Well, they did it by collecting data in a very flexible way, and in particular analysing their data in a very flexible way. And what they showed in their paper was an abstract that could be presented in a redacted form to give just what looks like a conclusion that you might want to sell to a journal, so to speak, and then the unredacted form, which included all of the different possible permutations of their data that they could explore. And it looks like this. I'll give you a moment to read it. So in bold is the redacted version, and then you have the unredacted version interspersed there as well. So the point here is that if you collect enough variables and you test for enough outcomes and you adjust for enough covariates and you try all of those different permutations of your data, you'll eventually arrive, quite quickly in fact, at a p-value less than 0.05. But of course that p-value doesn't mean what it was originally intended to mean when it was developed by Fisher, for example, 100 years ago. Because you've not accounted for the denominator, if you like, the number of times that you've rolled the dice to arrive at that conclusion. So what's shaping that kind of behaviour? Well, that's a difficult question to answer with respect to a single factor that is contributory. But if we look at the literature as a whole, we can see that there is actually quite good evidence that this flexible analysis is commonplace. So here you have a paper by Joshua Karp looking at the functional brain imaging literature in humans, where he reviewed about 240 different fMRI studies. On the right-hand side are different measures of reporting quality, and the first point here is that actually there often isn't the level of detail reported in the papers to allow you to fully reproduce the methods that were part of that experiment. And part of the constraint on how much we can report in papers is due to word limits, length limits, these anachronistic hangovers from our model of scientific communication, which is that we disseminate our findings on dead trees. When you look at the analysis pipelines in these 240 odd papers, you find that there are almost 240 unique analytical pipelines. fMRI is one of those technologies that provides you with a very large high-dimensional data set where you need to make lots of analytical choices as you work through that data set. And that means that the analytical space that you can work within is very large. And the fact that there are almost as many analytical pipelines as there are papers, in other words, almost no two papers analyze their data in the same way, even though they're using the same technology, indicates the degree of flexibility that's possible within the literature, but also the extent to which that flexibility is actually being leveraged to arrive at conclusions that are publishable. So this has been described by the blog in Euroskeptic and also by Andrew Gelman at Columbia as the Garden of Forking Paths problem, that we start with our data, and because we're scientists, we're interested in exploring our data. And of course, there's value in exploring data. And we take different steps down these different paths through our data. We take a few missteps, we retrace our steps, we try again, until we arrive at this p-value less than 0.05, which in many fields, particularly in the biomedical sciences, essentially is a criterion for publication, and by extension a criterion for reward and promotion and, cocktail parties and all the rest of it. But of course, that single test that we arrive at is predicated on you only having done one test. So if you don't declare the denominator, the number of forking paths that you went down, before you arrive at that p-value, then all bets are off in terms of interpretation. And so what we end up with in the literature is essentially exploratory research, which is presented as confirmatory, and which therefore is very difficult to reproduce. I'm not saying that there isn't value to exploratory research. Of course there is. We need to be pushing the boundaries of what we know to move into new areas. But when we're doing confirmatory research, we should be clear that it is confirmatory and conduct it accordingly. And when we're doing exploratory research, we should be clear that it's exploratory and describe it accordingly. Because if you look at the psychology literature, and again, I don't think it's particularly unique in this respect, we must be really good at hypothesizing or have some uncanny ability to predict the future, because all of our experiments work. Over 90% of the published psychology literature finds what it purports it was looking for in the first place, which suggests that either we are testing really mundane hypotheses that are really trivial, because they always pan out as we expect them to, or that we have this uncanny ability to predict the future. This is a really nice satire, actually. It's only short. If you've got a few minutes, you should read it. Or that something else is going on. That we are incentivized to create a narrative in our studies, to remove the messier data, present the cleanest trajectory from what we describe as our hypotheses, but they may not have been the original hypotheses that we had, through to our conclusions. And so you create this problem known as harking, hypothesizing after the results are known. Now, of course, it's fine to create new hypotheses after you've looked at your data, but what you shouldn't do is present your data as if they were hypotheses tests of a priori hypotheses that were in fact post hoc. So what evidence is that there are structural issues that contribute to this problem? Well, one of the cultural differences between different scientific ecosystems, if you like, in different countries is the extent to which scientists are directly rewarded for publication, for getting grants and so on. So one of the key differences is that for the most part in the UK, academics have their salary guaranteed irrespective of their grant income. And they might have their head of department come and knock on the door and have a sort of stern discussion without coffee if they don't get much grant income. But their salary doesn't directly, proximally depend on it. Whereas in the US, certainly in biomedical departments, a proportion of your salary, up to 100% in some medical schools, will have to come from grants. And if you don't get grants, you don't get paid. And when we look at the extent to which that is then reflected in patterning of the extent to which effect sizes in the published literature are overestimated relative to what we think are likely true effects, we see this kind of effect play out. Together with a colleague, Jonathan Flint, a few years ago, we showed this for genetic association studies, and that was replicated by Finelli and Ioannidis more recently, showing that certainly in the softer sciences, US studies tend to overestimate effect sizes relative to studies conducted in the rest of the world. Possibly because of different incentive structures in different countries. What other incentives? Well, if we're incentivized to generate lots of papers, and in particular to try to get into high-profile papers, should we put all of our eggs into one basket, use our limited resources to run one large, weighty, authoritative study that will definitely give us the right answer, but where that answer might be quite boring because there may be no effect, the treatment may not work, or whatever it might be? Or do we slice our resources into smaller and smaller amounts to run more studies, roll the dice more often, in the hope that we'll be able to publish more papers because we can analyse each of those studies relatively flexibly, but also that the chances of us finding something particularly eye-catching will be greater because many of those small studies will have a chance, at least, of identifying spuriously a very large effect. And when we looked at one literature, the neuroscience literature, but again, there's no reason to think that this is particularly unique in this respect, we found that the average power, the average ability of these studies to detect a true effect, if an effect is in fact there, was about 20%, whereas conventionally the power of a study is meant to be about 80%. So it was far, far smaller than it should be. These studies are too small, in most cases, to give reliable results. And it's plausible, of course, that that same problem exists at a higher level in that the funding councils, and they have systems in place to try and protect against this, but of course, in some countries at least, the political masters of those funding councils want to know how many groups have you funded, how many grants have you funded. And the more you can say you funded, the better it looks, because you're funding lots of labs across the country and generating lots of research activity across the country. But of course, what you're doing then again is thin slicing your resources to generate more coverage, and those individual labs are then thin slicing their resources into multiple studies, potentially, which just exacerbates the problem. So what can we rely on? Can we rely on the journal that a study is published in? Can we rely on the institution that generates the research? Possibly not. Malcolm MacLeod at Edinburgh, who gives a far better version, more entertaining version of this talk than I do, looked systematically at animal studies and the quality of reporting of various measures in those studies that should protect against different biases and ensure the robustness of the research. Now, the fact that these measures aren't reported doesn't mean that they weren't conducted, that the studies didn't actually use these measures, but all we have to go on is what's reported in the paper, so this is the best that we have. And he showed that, first of all, many of these measures were simply not widely reported: randomization, blinding, sample size calculation in these animal studies, these things that should ensure the robustness of the findings. But critically, what he found was that when he looked at the top ranked institutions in the UK based on the last ref, those studies did worse on these measures of reporting quality than a study drawn at random from PubMed. So our best institutions are actually doing worse than we would expect just on the basis of any study in that particular area drawn at random from PubMed. We've also shown separately that things like journal impact factor are a very poor measure of quality, and in fact the impact factor is a stronger predictor of the likelihood of retraction than it is of the number of citations that an article gets. So we blithely assume that this is all fine, that of course the process of science is messy, which it is, but that ultimately science will self-correct. These false starts will eventually peter out, these rabbit holes will come to an end, and we'll retrace our steps and go off in another direction. And of course that's true if we apply the scientific method correctly. But it's an empirical question, in my opinion, the extent to which we are applying the scientific method correctly, the extent to which we are, for example, attempting to replicate initial findings to confirm that they're robust. So I don't think we should blithely assume that science will sort itself out, because A, it might not, at least within a reasonable time frame, and B, even if it does eventually, maybe we could reshape the system and reshape the way that we work to make sure that it does so more efficiently. Because if you look at the literature, you find that replication is the exception rather than the norm. So this is a study recently in the British Journal of Psychiatry which looked at 83 articles recommending an intervention. So these were clinical trials which, on the basis of the results of that single trial, explicitly recommended a particular intervention. But about half of them hadn't been subject to any attempt at replication. And of those that had been subject to an attempt at replication, which you can see on the right-hand side, a proportion were indeed replicated, that's the far right bar. A good proportion found that the initial effect that was reported was much stronger than that which was indicated by the replication study, this regression to the mean of effect sizes that we see pretty much whenever we look for it, that these initial studies tend to overstate how strong an effect is. And a good proportion were actually contradicted, failed to replicate. And these are studies that are explicitly recommending an intervention. So there isn't enough replication, and when there is, we find that actually many of our findings are not as robust as we might expect them to be. And one of the difficulties here is that scientists themselves, despite being trained to be this Platonic ideal, are human and therefore subject to all of the biases that anyone is. And in particular, that we have our own pet theories, vested interests. They may not be financial vested interests, but they are personal vested interests. And that was shown quite nicely in this study, where a set of ambiguous results, the sort of thing that could be interpreted as supporting a hypothesis or not, depending on what side of bed you got out of, was presented to two groups of people: methodologists who had no skin in the game, no investment in that particular research field, and people who'd published in that area. And the people who'd published in that area were much more likely to interpret these ambiguous results as supporting their hypothesis than the methodologists. In other words, your personal investment in the field shapes your interpretation of data. Unconsciously. Most of these biases, these behaviours that I'm talking about, operate unconsciously. That's why they're interesting to psychologists. These aren't people who are actively going out to try and game the system. Quite the opposite. These are people just going about their business, trying to use the resources that they have to the best of their ability to discover new things, but doing so in a way that doesn't protect them from themselves, if you like, and from these unconscious biases that affect all of us. And the problem that this creates is that if you come into a field as a new or an early career scientist, you can get a very distorted impression of just how robust that particular field is. So this is a study from a few years ago in the British Medical Journal, looking at citation networks. And the specific topic doesn't really matter for the purposes of illustration. What you have on the right-hand side are studies published in two linked research fields. You have the primary data on the left-hand side in white circles. Year of publication is on the y-axis, going from the early 90s to the early 2000s. And you have 6 studies that were critical and 5 studies that were supportive. And those supportive studies were sufficient to generate the next stage of research, if you like, which was the animal cell culture research, which are the blue circles. And if you look at the citations from that animal and cell culture research back to the original data that supported the genesis of that field, if you like, all of the citations are to the supportive data. There are no citations to the critical data, except for one citation, which is in white because it was a critical citation, saying, well, there's this study, but it's a bit crap. So if you're a new researcher coming into that animal cell culture field, it would look like that literature is built on a really robust foundation because all of the evidence that's cited in support of it is supportive. But there's this other hidden literature which is invisible. It's hard to find because it's not being cited by anyone. You'd have to do some really serious due diligence to dig that literature out. And only then would you have a more balanced sense of how robust the foundation of that literature actually is. And we've done something similar ourselves, looking at another literature, in this case the serotonin transporter gene and amygdala activation, but again the topic doesn't matter particularly. And we found 2 things. The first is that there are many studies that make a claim in the abstract which is often not supported by a more sort of impartial reading of the actual data in the article. So there are some studies that are positive, that show an association in this case. There are some for which the evidence is more ambiguous, but the authors are optimistic in their interpretation in the abstract. And then there are some that show no association. And those proportions are shown in the inner part of that donut. The top one is the Web of Science, the citation network within that particular literature, and the bottom one is the wider citation network across Web of Science. But on the outside, you have the number of citations that those articles are garnering. And the most striking finding is that the articles that describe bad news, there's no association, we didn't find anything, receive far fewer citations than would be expected based on the proportion of studies that represented there. 21% of the studies within the network don't show an association, but they only get 3% of the citations, and in the wider Web of Science citation network, the figures are 25% and 4%. So people just don't want to cite bad news, basically. So again, you get this very distorted sense of how robust the literature is. So is science self-correcting? We can certainly introduce measures to help it to self-correct. So here you have data from clinical trials funded by the National Heart, Lung and Blood Institute in the US. And the little flag there in 2000 was the date when they required the registration of primary outcomes on clinicaltrials.gov. Before that, nearly all studies showed a benefit of the intervention over placebo on the primary outcome as described by the authors, but there was no way of checking that that was actually the a priori primary outcome. Once they had to pre-specify what their primary outcome was, the vast majority of studies were null. They showed no benefit of the intervention over the control. Because science often is quite boring. The answer is often no. There is no effect. And that's just the reality of the situation. But again, we're not particularly incentivized to publish or to disseminate those kinds of findings. And in fact, strikingly, there was only one study that showed a harm of the intervention over the control condition, and that was only observed after the pre-registration of primary outcomes was mandated. So that's the reason why I describe the environment that we work within as a scientific ecosystem, because we're all trying to be good scientists, but we're also at the same time human, and we're incentivized to behave in certain ways. And the ways in which we're incentivized may have become distorted over the years so that they're no longer aligned with what's in the best interests of advancing knowledge. I've tried to model this with an evolutionary biologist who was at Bristol, he's now at Exeter, Andy Higginson. And this is essentially a figure showing the fitness function of scientists. So if you imagine scientists as little furry creatures foraging for interesting results that they can then go on and publish and use to generate career advancement. We calculated a metric for the sort of benefit to science of the work that we do, where red is better and blue is worse. And it's a multiple of the proportion of exploratory studies that you do, because you need to have some exploratory work, the proportion of confirmatory studies that you do, because you need to confirm those initial findings as well, and the proportion of those that's actually correct, because of course there's no value in generating false findings ultimately. And the position that we find ourselves in at the moment is up here, when you consider the extent to which there is a diminishing return to publishing additional papers and the weighting that's given to exploratory results. If you look at the structure of the ref, for example, we're asked to provide our 4 best papers, where best nearly always means novel and exciting. So we're in that top right-hand corner. We're incentivized to produce a relatively small number of papers that are very eye-catching, groundbreaking, novel, exploratory, and so on. And that puts us in the top right-hand corner. You see similar structures in the US when people are applying for promotion, and in the promotion pack, when you have to write a letter of support, you get their 4 best papers or their 5 best papers. So there's this focus on, and it's understandable why you would want to focus on quality over quantity, where quality is defined as novelty and eye-catching findings. So what can we do about that? Well, one option is that we can move the red area up to where we already are. Journals, for example, can require more stringent statistical evidence before they allow work to be published, or require that studies are larger, and both of those will serve to push the red up to the position where we already are. Or we can move our incentives into where the red is without changing what journals do, for example, by giving more weight to confirmatory studies, in promotion decisions, in the ref, in hiring decisions, and so on, and also take into account the volume of work, particularly if a good proportion of that work is confirmatory, and try to move away from this focus purely on publishing in certain kinds of journals, in my field, Science, Nature, Cell, Neuron, that kind of thing, very high impact, but where they explicitly focus on the high risk, high return findings. And the editors are quite clear about this. We're looking for the really exciting, eye-catching stuff which, if correct, will be transformative. But we accept that, by definition, much of that work won't be correct because we're taking a risk by doing that kind of groundbreaking work. Now, there's a place for that. That plays an important part in science, but we shouldn't be focusing all of our effort on high risk, high return. That feels a little bit too like a banking crisis waiting to happen. So what can we do about it? Well, I've already alluded to the fact that there could be solutions from the bottom up and from the top down. Funders, journals, institutions can mandate ways of working, but there can also be solutions from the bottom up. Individual research groups can start to work in ways that improve the quality, the robustness of what we do. And so we've tried to do this in my group by increasingly adopting an open science model to the way that we work. And it's not a trivial process, because you can't suddenly start making all of your data open, for example, if your participants in your research haven't consented to that. So you need to change your consent procedures. You need to work with your research governance team. And we've done that over the years, so that now we pre-register all of our studies, so that we pre-state our primary outcomes and our analysis plans in advance. And we do that all the way down to undergraduate projects. We consent our participants so that the data can be made open after the study's complete, and we've just started the process of doing that. And because we are largely funded by the Medical Research Council, all of our publications are gold open access, but even if they aren't, we can make them green open access. So we describe that process in this commentary here. And there's good evidence that that actually makes a difference. For example, there's this nice paper here showing that the willingness to share research data is related to the strength of evidence and the quality of the results reported. Because our experience has been that when you have a postdoc, say, or a PhD student who knows that their data will be made open, they just check everything a 4th time or a 5th time. It serves as a quality control measure. It's not that realistically we think that hundreds of people are suddenly going to start downloading all of our data files and probing them for errors, because frankly people have got better things to do. But the fact that they could, if they wanted to, just act as a sanity check on our excitement when it comes to writing up results that we think look really interesting. So everything just gets checked a little bit more scrupulously. Because the reality is that in any human endeavour, there's going to be human error. And the fact that there are articles out there that have been reviewed by 2 or 3 people 2 or 3 times, that have been read by all of the authors, that have been proofread, copy edited in some cases, and there are still typos in them, suggests that we're never going to be able to get rid of human error completely. But what we need to do is to introduce processes that mitigate that minimize it as much as possible, but also accept that it inevitably will happen, because if there are typos in published papers, there are going to be typos in data sets. And those typos will hopefully, most of the time, make no difference to the results or the conclusions drawn from that data set, but they will exist. And at the moment, we come down very hard on academics who make mistakes, honest mistakes, not consciously, but there's a sort of embarrassment associated with having to issue a retraction notice, even if that retraction is due to honest error. And there shouldn't be, because actually we should be encouraging people to identify their own errors in the same way that the airline industry encourages pilots and in fact anyone involved in the aviation industry to speak out if they find anything that they think might have an impact on the safety of the aeroplane that's about to take off. So they create a much more kind of open and just culture around reporting of errors. And I think that's something that we could learn from, because I think that would be a healthy culture to adopt within academia. We're working with a funder in the US, the CHDI Foundation, that funds Huntington's research at the moment, and we published this commentary that came out of a working group that we convened to discuss how funders could change the way in which they support research to improve quality control. And the analogy that was drawn by Roby Blumenstein, who was the head of this organisation, was that the situation in scientific research at the moment is much like the automobile industry in the US in the 1970s. At that time, the US automobile industry was focused on productivity, generating lots and lots of cars. They'd all roll off the production line very quickly. Someone would count the wheels, check the engine started, they'd drive off, and they'd break down a few weeks later. It was the era of the lemon. So I gave this talk to some students, and no one knew what a lemon was, which tells you a lot about how much more reliable cars are now. But hopefully some of you in the room will remember what lemons are in the automobile context. So, these were cars that were built to be fixed later. A statistician, Edward Deming, in the US, went to Japan and introduced quality control measures at every step of the production pipeline. So you build a small part, you check that it's well built, you put it into a larger part, you check that that works. Eventually you have a car. And the Japanese automobile industry still has a reputation for reliability 50 years later on the basis of those changes to quality control at every step of the production pipeline. And the critical insight was that by doing that, by introducing quality control at every step of the pipeline, not only do you improve the quality of your product, but you also improve productivity, because the whole process ultimately becomes more efficient because you don't have bits breaking down all over the place. Everything just runs more smoothly. So it's not just that you produce better work, but you can produce it faster and more efficiently in the long run as well. So I think there's a lot that can be learned from the way in which the automobile industry changed to adopt these practices, and we can implement a lot of that in biomedical research and probably more widely as well. And I think open science has a critical part to play in that by introducing processes that implicitly bring quality control measures into our daily practice. And I think open data is a really good example of that. Because the really striking thing about the psychology reproducibility project were only about 40% of those studies that were subject to a replication attempt. They took 100 studies from 3 psychology journals published in a given year. Only about 40% could be reproduced with a p-value less than 0.05, was that when people were asked to bet money on which studies would replicate and which wouldn't, in a prediction market where you could buy or sell positions based with real money, because this was economists doing this study and they give you real money to work with, based on whether or not you thought that finding would replicate, the market very accurately predicted what actually happened. So the red squares are failed replications, the black squares are successful replications, and you can see that the market, black dominates in the top right, where the market would have predicted replication, and red dominates in the bottom left, where the market would have predicted failure to replicate. So we knew, as a community, we had a gut feeling about which studies were robust and which weren't. But if you were a new early career researcher coming to this field, you would have no idea, because this is the knowledge that's held privately, if you like. And the experience that many PhD students have, myself included, is that you go into a field all wide-eyed and optimistic, and you build this proposal, this program of research, on this foundation that you think is robust because every study that you read finds the effect that it should be finding. You run your first study, and it fails to replicate. And so you have this, or it fails to show the effect that you're expecting. So you have this crisis of confidence, and you think, I'm obviously a terrible scientist. But actually, you then go to a conference and speak to this grizzled old academic who goes, oh yeah, everyone knows that effect's much flakier than you'd think from reading the papers. That person didn't know. So we need to make all of that knowledge much more open. So we need to, again, think about the way in which we do science, because the way in which we do statistics, null hypothesis significance testing, is 100 years old. The way in which we disseminate our ideas via dead trees, I hope that's not offensive to anyone in this audience, is 400 years old, the academic journal. So maybe it's time for an update, in particular because we now have statistical software packages that allow us to run hundreds of tests, whereas a few years ago we'd have had to have done it all by hand. We now have indexing services that allow us to search hundreds of abstracts without ever having to actually read the paper. So this acceleration, which is focused on productivity, arguably comes at the expense of scholarship and potentially quality control. And so we need to build in new processes and new systems that allow us to just refine the way in which we do science and ensure a higher quality product at the end. Because these aren't new problems. I'll just end with this quote from Babbage in 1830, Reflections of the Decline of Science in England, and I'm sort of planning to write the sequel when I retire. This is an art of various forms, the object of which is to give ordinary observations the appearance and character of those of the highest degree of accuracy. One of its numerous processes is to make multitudes of observations, and out of these to select only those which agree or very nearly agree. If 100 observations are made, the cook must be very unhappy if he cannot pick out 15 or 20 which will do for serving up. The problems are not new, but the scale of the problem, the speed with which these problems become realised, the scale in terms of the number of groups around the world that are working on these questions at the moment, arguably has changed. And so perhaps we need to think about updating the way in which we do science to accommodate that. Thank you.